person can do that on its own.
So I hope you've all digested what we covered yesterday, and so that you can start on a new topic again. And the first part of this morning, I'll try to walk you through randomized trials, which is one of the trickier designs to carry out, at least, and probably not so much to design. But basically, to start with a simple definition, randomized trial is a study where people are divided into groups using random allocation. So they either receive an intervention or they don't. And so there are comparisons to the experimental, the new intervention, and then either nothing or an existing, well-known, established intervention. And according to the International Dictionary of Epidemiology, there's a bit longer definition that's shown on the bottom of the slide, but basically it just states the same. And the virtue, the major advantage of a randomized trial is really in the randomization procedure. The fact that it is the only means that we can more or less guarantee that we have two comparable groups, so that any differences between them during the follow-up will be due to the intervention, but this is provided that we can maintain the similarity. It is not so difficult to create groups that are comparable, but the challenge is usually in maintaining the comparability during the trial. And also, of course, really keeping the allocation in the sense that everybody who's allocated to an intervention really receives it. So these are the two major challenges for randomized trials. Okay, so just to go a bit deeper into the basic issues. So we have a chance mechanism, and the idea is that there should be a similar probability of assignment to each subject. This doesn't mean necessarily that everybody has a 50-50 chance. So it could be also something like 1 to 2 or another kind of probability, but still for each subject. And in some cases, there could be even divided into strata, into groups, so that there is similar probability. Another major issue is concealment, which means that we are sort of making the allocation unpredictable, so that there's no way for anybody to be able to guess in advance what will be the outcome of the allocation. And okay, I won't go into the details of the techniques. So, as I said, we have an intervention group, an experimental arm of the study, where people are allocated to receive what we are studying. It is the exposure that we usually only observe, but in a randomized trial, the investigators produce the exposure. So there's more control over the exposure. It can be designed so it will be ideal in terms of the administration, dosing, duration, and so forth, which is not the case in other study designs where we are simply recording what has taken place. And we need a comparison, a reference, which would be the control arm that does not receive a similar intervention. So there could be a reference, current best practice typically in a treatment trial, and perhaps no intervention in a screening trial, or screening using another test, or a later start of screening, less frequent screening, or some other procedure. If there is no intervention, then it's common to use a placebo, which means a sham intervention, something that appears to be an intervention, but it's thought to be completely inert, inactive, so it would not produce any effect, but it's just for the purpose that the people in the trial would be unaware of whether there was an intervention or not. As I said, there's a minimum of 2 groups. There could be several. It would be more complicated, for instance, 2 new interventions or combinations of interventions that are compared. And then during the course of the study, we follow up, as in a cohort study, the study groups and record the outcome events as they occur. And one of the things that I've learned from Olli Miettinen's teaching is that the most important aspect of study design is the design of aims. That is, carefully deciding what is really the aim of the study, as specifically as possible. And for a randomized controlled trial, this is even more important than for other study designs, because there can be only a single well-defined objective. And the PICO way of summarizing it can be used also for randomized study. The other key features besides the aim are, as I already mentioned, random allocation, allocation concealment, and blinding, which will be covered in some detail during this talk. So the PICO summary, as was mentioned yesterday, we have the population for P, who are the subjects. That's defined by the inclusion criteria, the exclusion criteria, who are eligible to be included. The intervention, the experimental procedure, treatment, how it is conducted, when, where, by whom, these are all things that need to be very carefully planned so that we really can provide an answer to the question. If this is not optimal, then failure to produce a different could be just due to the fact that the experimental procedure was not planned well enough. The comparator I already mentioned, so it is the control group. What do we give them? It is important in order to have a meaningful research question. Of course, the questions in any study should be, how can we improve from the current practice? So therefore, the reference group should be something that would be equivalent to the current practice. If there is already a well-established treatment with known effectiveness, then it does not make much sense to conduct a study compared with simply placebo, because that would not inform us about which is superior, the new experimental intervention or the current practice. And just if there is already a well-established effective method of treating or screening, then just being able to tell that there is another one also is not really useful. But really we're comparing head to head the current best with the new intervention. And also there are various study designs to assess whether we are looking at superiority, whether there is the new treatment is better, or whether it's similar, or whether it's non-inferior or it's not worse than the existing one. And so this is also something that needs to be borne in mind when planning a randomized study. Outcome, this is, as always, should be something that is really the aim of treatment. Are the patients cured? Do they survive? What is their functional status, perceived health, quality of life, something really relevant for the patients, and not only some paraclinical parameters such as blood tests or X-rays or whatever. Indirect measures of disease activity that may have little bearing to the patient's well-being. And just took the classical example, the UK trial of Streptomycin for tuberculosis is regarded as the first modern randomized trial. So applying the PICO summary to it, the patients were subjects with smear or culture-positive lung tuberculosis. The experimental intervention was intramuscular streptomycin, 500 milligrams, 4 times per day. Comparator was the usual care at the time, which was simply bed rest. Nowadays, that would be completely unacceptable. The outcome was mortality at 6 months, and as you can see, 7% and 27%. First of all, the level of mortality was exceedingly high by current standards, and the treatment effect was also huge. 20% reduction. NNT, as you can already calculate in your head, is 5. Okay, and the conduct of randomized trial, we start from the eligible population defined by the criteria, inclusion and exclusion. We recruit the people, we seek their consent, and then through these two mechanisms we come up with the study population. Then we generate random allocation, which creates the 2 arms, and then we follow up for the outcomes. And what makes this a randomized study is this process in here. Of course, here between the allocation and follow-up, there should be the intervention, administering the intervention, providing it for the study subjects. And even more historical example is James Lind's treatise of the scurvy, 1747. Really a classic. The study design was described here. Scurvy was a major disaster in the English Navy and all other navies when the sailing ships, they took months in their voyages, and so the diet was very deficient, and they had no understanding about what caused scurvy. And so James Lind, I don't know how he conceived these 6 different interventions, providing either cider, barley water, sulfur acid, I would not have wanted to be in that group, vinegar, seawater, or citrus fruit for the subjects. And I don't think the study had adequate statistical power, but what was observed that these 2 patients recovered while there was no real change for the others, and the outcome was that, okay, they should carry citrus fruit aboard the English ships. Okay. So randomized trial is always a major undertaking. The preparation phase takes months in the minimum. Years is not uncommon to get all the permissions, to write a proper study protocol, to obtain funding and recruit subjects. It's thought to be the best means of improving our understanding, the most rigorous tool for knowledge acquisition, strongest evidence for causality in medicine. And the idea there is that if we think of causation as production of outcomes, then in a randomized trial we are administering the exposure, the intervention is thought to produce the outcomes. So it is more concretely, more tangibly observable, the causation, which usually is only a product of data analysis and very indirect inference. So in a randomized trial we can be assured that the effect that we observe is truly attributable to the intervention, provided that we are really conducting a high-quality trial. And basically, the researcher has the capacity to manipulate, to interfere with the natural processes and really produce the results of the trial. And CONSORT is Consolidated Standards for Randomized Trials. So it's a reporting guideline. And here is the standard CONSORT diagram, which describes how one should report the results. So numbers of people that are assessed for eligibility, exclusions, number of people randomized, numbers of people allocated to the 2 interventions, numbers of people who received or did not receive the planned intervention. These should not exit the study, however. Then some people have always lost to follow-up of people, and then finally the number of people in each arm analyzed. And of course, the number of people included in the analysis should be as close as possible to those number of those randomized. Any loss, dropout during the study will weaken not only the statistical power, the size of the study population, but also causes potential bias. As rarely the people who exit the study are similar to those who remain in it. They could drop out due to adverse effects or lack of therapeutic effectiveness or poor health or other reasons, and they tend to do worse in many respects typically than those who remain in the study. Just keep repeating this point because I think it is really important. So random allocation, its chief virtue is that it maximizes comparability of groups. I cannot say it guarantees because it is a stochastic process, but it maximizes the best means of achieving comparability. And most of our inferences about the reasons for differences between the group rest on an assumption which is called in Latin ceteris paribus, all other things being equal. So John Stuart Mill already proposed a model which is called single difference model of causation, so that if something occurs in one circumstance and not in the other, and there's only one difference between them, then the difference is the cause. And this is the rationale of randomized trial and also the counterfactual model, so that we try to construct comparisons where any difference between two groups in the outcome would be attributable to the exposure of interest, the intervention in a randomized study, and as I said, the random allocation is the best means for providing such ceteris paribus comparability. Randomization eliminates or minimizes the bias from treatment assignments. And therefore, there's no selection bias, meaning that the groups really are similar to start with at least. However, it's important to bear in mind that we need to remember the law of large numbers, meaning that if we randomize only a handful of times, then the probability of having similar groups is very small. Only through large numbers of repetitions we acquire the stability to the randomization. And another beauty that's not achievable by any other means is that not only does randomization create similar distribution of known confounders, it can also be safely assumed that it will also diminish all differences in unknown determinants of the outcome. So if there are risk factors that we have not identified yet, then in a cohort study or case control study, there's no way of assessing even whether there are differences. However, in a randomized study, we can safely assume that by virtue of randomization, there will be minimal differences also in those factors that have not yet been identified. Which means, once again, that the ceteris paribus, the comparability, is optimal. And once again, I try to underline the importance in the bottom line that the maintaining the comparability is a major challenge for randomized trial. So it requires that the study is executed flawlessly from the design through the conduct of the study to the analysis and reporting. Just by doing randomization and then not paying any adequate attention to the conduct of the study will not give reliable results. There are also certain limitations for starting a randomized study. One of them is related to the knowledge status, and that's called Equipoise, another fancy term that I think Malcolm said that we can use to sort of get dominance over the readers or reviewers or whatever. It just means that there is a genuine balance of uncertainty about the superiority of intervention, so that we don't know whether the new intervention that we are evaluating, whether it's as good or worse or similar to the existing approaches. So meaning that there is up to that point insufficient evidence to tip the scale in favor of either option. So we don't know really. The question is open that we are looking at. Is this new proposed method, is it superior to what we already have? You could call this, once again using philosophical terminology, epistemic uncertainty. The knowledge is not complete, far from it. Because in order to conduct a study, to expose people to willful manipulation, we really have to be able to obtain new knowledge from the study. Otherwise, the risks are not worth taking. So this means that there's a balance of chances about the results. It's equally possible that the new treatment will turn out to be better, but also that it will not. And that leaves us a window of opportunity. We have to have some kind of preliminary basis for assuming that this might be the case, that this is really a promising treatment or intervention, but it's not yet fully established. So not too early, because we need the justification, and not too late, because if the question is already settled, then conducting a trial is no longer feasible. So we have to have sufficient grounds to believe that there might be an effect. There should be some what's nowadays called proof of principle studies that show some effect in some preliminary setting, maybe an animal model or some basic science mechanism of action is well established, and perhaps some phase 1-2 data about safety. And the reason for not being able to conduct randomized trials when we already know is, of course, ethical. It's not possible to deny patients or subject an intervention that is known to be effective by randomization to something that we know is inferior. So if we have a new treatment, and there are already several trials that show it's better than the older alternatives, then conducting one more trial just in order to do science, get a publication, is not justified, because then we will be randomizing patients, let's say 50-50, to the new but already established superior treatment and the older, worse treatment, and the latter group would not be ethically justified. And the ethical issues are vastly more important in randomized studies than in non-randomized, non-experimental studies, just by the fact that we are responsible for administering the intervention. And if there's any adverse outcomes, then that is due to something that the investigator had created, produced, which is different from a normal non-experimental study where we are just observing something, and if you find out that something that took place increased the risk, that is not the responsibility of the researcher. But the ethical basis of clinical research and randomized trials is in the Helsinki Declaration, and one of the basic tenets is that treatment of patient must not be jeopardized, so that they should always be the first concern of the attending physician, that the patient would get the best possible treatment. And placebo is unacceptable by current standards whenever an effective treatment exists, and therefore the comparator should be an active treatment, the standard treatment, if there is established practice guideline, for instance. The intervention really creates the responsibility for safety of the subjects because there is manipulation, and it creates always a possibility of harm, what would be maleficence in the ethical basic principles. Unintentional harm from intentional manipulation. So we should have a good understanding about the possible risks whenever starting a trial. And the ethical committee requires that we can really claim that the potential harms are balanced with potential gains, meaning that the risks are not so large that they would threaten the importance of the improvement in treatment outcomes. And also, if we are going to recruit patients to a randomized study, informed consent is absolutely essential. The researchers must provide sufficient amount of information about all of the major issues, such as that, first of all, that this is a randomized study, this is science, this is not just routine patient care, about what is the research question, what are the inclusion criteria, how many patients will be included, what is the funding of the study, who is responsible for conduct of the study, and all these details about the study, in such a way that the patient can understand them, so that in the language that they can understand, and in terminology that is understandable for the patients. And this can be very demanding, for instance, when we are talking about pediatric patients, and there you might need to even design several information letters for children of different age. And there are some special groups where it's not really clear whether they are completely autonomous and competent, such as prisoners or expecting mothers or people with mental illness or handicap. and they require even more stringent scrutiny by the ethical review board. Conducting a randomized study is very demanding, so the logistics are complex. Just writing a protocol is a huge task. It needs to be very exhaustive, very detailed. Every possible detail has to be thought through and outlined in the protocol. Eligibility and exclusion criteria are just a start, but also the procedures for conducting the intervention and documenting the intervention, but also the enrollment stage, the follow-up stage, and data analysis. Those should all be already outlined in the study plan. There are financial issues often involved. How about the health insurance for any potential adverse outcomes? Are there any fees for participation, compensation for travel, time, and so forth? And there are local regulations about those. Safety monitoring. Typically in a randomized trial, you would need a committee external of the researchers who would have access to the data to evaluate whether there are any serious adverse events, and possibility of stopping the study. Having clear stopping rules, when should the study be stopped, is also important. The permissions, evaluations, reviews prior to start are an effort. And often large sample sizes are needed, and the recruitment goals need to be realistic. It's far too common for trials to fail because they have set too optimistic recruitment goals, and when it turns out that they cannot be met, then the study just fades away. And it's not something that you can start just any time. You need to collect all of the issues that are needed: the money, the time, the manpower, expertise, and trained staff to conduct a randomized study. The first set of limitations of the trials has to do with generalizability, meaning how applicable are the results in other patients than in the trial. External validity sometimes. And the main issue here is the exclusion criteria that are often very detailed, long list of prohibitive issues. And the starting point is the primum est non nocere, first do no harm principle already in the Hippocratic Oath. So basically the rationale is that we should try to exclude anybody who is thought to be at a higher risk of the adverse outcomes. So typically patients with almost any comorbidity are excluded. Too often any women of childbearing age, for the rationale that they might be pregnant, which will already distort the patient population. So the outcome may finally be that the results are not readily generalizable to all of the patients. In real life tend to have other diseases than only single one. In trial patients tend to be younger than, for this reason, than the average patients, and this might be an issue. The comparator I've already mentioned. So how do we treat the reference group? Patients, subjects in the control arm. Is there no intervention, or just a placebo, or a standard treatment? And this is really important for the interpretation for the results. This defines really the study question. And as I mentioned, they should represent the management of the study subjects if they were not in the trial. Because the most relevant question for a study is really, does this new technique improve outcomes compared with the current standard approach? That would be the most important issue. And it's also, unfortunately, far too common to find, basically for pharmaceutical industry-sponsored studies, that they willingly use a comparator that is chosen so that it would make the new intervention look good. It could be too low dose, for instance, in the comparator group, so that they would know at the outset already that this will result in poor outcomes, which makes the new intervention appear superior. It could be too high dose, so that it would result in adverse effects and dropout. Or there could be placebo when there clearly is an established treatment, and then the result will be uninformative. Just that we have a treatment that's shown to be effective. Okay, now there's another one, but we don't know which one is better. Because, comparing between studies due to the different interventions, different selection criteria, and so forth is not straightforward. Randomization, I believe we've covered already in some detail. So basically nowadays we always use computer-generated random numbers. And the key issues in randomization is that it really must be unpredictable, and for computer that always is. There should be create a balance of key features between strata, so that this means that if we have, for instance, if we randomize by group stratified randomization, so within each group there should be similar distribution across the trial arms. It must be well documented so that even afterwards the reader and the reviewer can really be certain that it was done properly. And as I said, we need to bear in mind the law of large numbers, which means basically that when a random sample is drawn from a large population, with increasing sample size, the average of any characteristic will be closer to the expected value. Meaning, in this case, that when we repeat the random allocation large enough number of times, then the similarity between the groups increases. Allocation conceal already mentioned also, so that meaning that the rationale is that there should be no possibility for the people enrolling the subject to know what will be the allocation. So the assignment sequence must be such that you cannot predict what will be the outcome. So if you, let's say you have a patient you sympathize with and you firmly believe that the new intervention is superior, then you would try to get this particular patient to the intervention arm, but the randomization should really be such that there's no way for you to be able to sort of manipulate the randomization. And also one of the good procedures in a randomized study is that there's separate, the allocation is done separately from recruitment. So those people who recruit the patients are not the same ones who do the allocation. But typically it's done centrally, the allocation at one single spot. And the treatment, the outcome of the allocation must not be revealed before the patient is in the trial, because that might really result in selection bias, so that the investigators and the subjects are unaware of what will happen. I already mentioned in passing sample size, and for sample size calculations to predicting the power of the trial, you need to have realistic estimates of the baseline risk. What is the event rate in control arm? So you have, for instance, how common are the outcomes in this particular group of patients, and you have long enough follow-up. And this is really needed in order to justify the effect size. And also you need to take into account the attrition, loss to follow-up, dropout that always takes place in a study, so it's not sufficient to just enroll an adequate number of people or to get consent, but also to keep them in the trial up to the analysis. Non-adherence will also dilute the results, and that needs to be taken into account also in calculating the sample size. And just a point that I think Tony Chen made very well yesterday about the statistical and clinical significance. So if we, for instance, say that the risk difference is minimum clinically relevant risk difference, let's say 0.1, then these are the results of some hypothetical trials. So the first one, and the statistical significance has to do with the breadth of the confidence intervals. So, for instance, this trial, where this is the point estimate, And is that the confidence interval is not statistically significant and it's not clinically significant. The second one here, this is the confidence interval is below the zero, meaning that it is statistically significant, but the effect size is only half of that deemed as minimal clinically relevant. So this is too small to really make any difference in practice. Here, the point estimate is below this point, but the confidence interval doesn't reach the zero level, but it's above the minus 0.1 level, so it is statistically significant, but it's not clearly clinically significant. This is sort of not statistically significant, but might be still clinically significant because the point estimate is exactly the 0.1. And here finally we have a result that is both statistically significant and it is also clearly larger the effect than what we have defined as clinically relevant. So this is important to bear in mind. And what should be, as Tony and Amy told yesterday, the important thing for you is the clinical significance much more than statistical significance. Placebo we use to reduce information bias, to improve comparability, because there is always an expectation of benefit, sort of a generic treatment effect. Some people call it doctor's face time, so the fact that you see the doctor frequently, and actually there are studies showing that if the doctor assumes benefit, then the patient will also improve from that expectation. And also there's what is known as Hawthorne effect. Just the fact that people receive more attention, they see the doctor frequently, they go to have tests more frequently if they are in a trial compared with if they are not. This might also improve the outcomes. So we use an inert so-called pseudo-treatment, and the idea is that it should be such that people are not able to tell whether they received the experimental or active intervention or the placebo. And for that purpose, we sometimes use a dummy, which is just a way of administering a placebo, so that if you are comparing a drug that is taken once a day, another one that is taken twice a day, then for those who receive the drug only once per day, then they would take a placebo, let's say in the evening, if they take only in the morning the active drug, so that the groups would be more similar in terms of the procedure of administering the intervention. And this also applies that, for instance, if there is potential known specific adverse effect of one of the treatments, then all patients in the trial should receive monitoring for that, not only those who actually receive it, because that will make, once again, a difference between the arms. And I've already mentioned the ethical requirements for use of placebo. Of course, if it's inert, it should be safe to the patient, and no intervention with established effectiveness is denied by use of placebo. And however, according to the current interpretation of the Helsinki Declaration, minor transient treatment benefit could be postponed. So if it's, let's say, eczema or something like that, then it can be then allowed to use placebo even if an existing treatment has been established. Blinding is another important issue in randomized studies, which means keeping allocation secret or unknown, so that both the participants and the researchers should be kept blinded in terms of the arm, the allocation, what kind of intervention is a specific patient receiving. And that should be during the intervention and during the follow-up. So sometimes we talk about single blind studies when only participants are not aware of the assignment, double blind when both participants and investigators are unaware, and triple blind when there's also a third group that is kept unaware of it. And the rationale is really to keep them comparable, keep the results comparable, avoid information bias, mainly due to differential assessment of outcome. And this is most important, actually not well for the patient, but also for the staff who makes the assessments, so that they would have no knowledge about the group of the patient that they are assessing. And this is mostly important for so-called soft subjective outcomes, when the outcome requires some judgment. If it's death, mortality, then actually it's been shown that blinding doesn't affect the results. But whenever there are an element of subjectiveness in the judgment, in the assessments, whether by the investigator or the participant, then blinding is important. There are studies are showing that the differences tend to be larger whenever there is no blinding. And in the reports of trials, they should be detailed enough to tell who was blinded and how was the blindness assured. And of course there are difficulties for blinding also. It could be impossible if the intervention that we are studying is something related to surgery or diet or exercise or education. However, for most of these we can also come up with something that we call sham or placebo. In my hometown of Tampere there was a trial of arthroscopic surgery for meniscal tears, or actually published 2 years ago in JAMA or Lancet or New England Journal, but they made sham surgery so that the patients were shown a video about an operation and the patient did not know whether it was their knee or something else. So there was a small cut made into the knee and they were completely unaware of whether they had been operated or not, but I think that's very avant-garde already, but it's possible. Similarly for diet, you could use something that is not really in accordance with the expectations. Education, you could educate people about something else than is really the study question. So it's possible, but it's difficult in many cases. There are these famous stories about how people would find out about the different drugs, about their smell or taste or feel or weight or whatever. For instance, and of course, side effects are also something that could reveal the allocation if they are really distinctive for an intervention. And basically, if you have a very effective treatment against a completely ineffective treatment, then the results might reveal the assignment trial arm. And also a colleague of mine who's now already passed away, a professor of epidemiology in Finland, was involved in a trial and then he said, well, I don't want to be taking placebo for years, and he took the tablet to a chemist and asked him to find out whether this contained any active substance or not, but that's not really acceptable even for epidemiologists, so not something that I would endorse. But as blinding is important, it should also be assessed. So it is a good practice in a randomized study to ask in the end all of the subjects, what do you think? Did you receive this intervention or that? And if they are correct clearly more than 50% of the time, then blinding has clearly failed. And you can also ask the people who are assessing the outcomes the same question. Compliance is another issue. So if everybody assigned to the intervention will actually receive, there is non-compliance, non-adherence due to refusal, but also the other way, non-compliance due to people who are not assigned to an intervention, they might receive it, which result in contamination or spillover. So these both would reduce the difference between the arms and therefore the apparent effect of the intervention. Finally, even if a trial has been conducted properly, there is a possibility of doing an improper analysis, and the golden standard for analysis of randomized trials is the intention to treat principle. It just means that whatever happens, it is only the randomization, the random allocation that defines the groups that are used in the analysis, maintaining the original allocation. So if people drop out of the study, if possible, they should be included in the study, in the arm that they were assigned to, in an proper intention to treat analysis. This is to be avoided per protocol or treatment received analysis because it violates the random allocation. Contamination also similarly should be included regardless of contamination. No compliance, similarly if possible, should be included in the analysis. Okay, what are the limitations of randomized trials? Well, I already mentioned it to start with. A randomized trial can only address a single narrowly defined question. In a cohort study, you can look at different outcomes of a given exposure. In a case control study, you can analyze several risk factors for a given disease. For a randomized trial, there is no more than one primary question. So therefore, you would say that it's sort of a blunt instrument. You put all of your eggs in one basket and hope that it will provide. And really, as I said in the early part of the lecture, the research question is important, so providing an accurate answer to the wrong question is not a major achievement. So that question should be very carefully defined. However, it's commonplace that a trial might take several years, so by the time it's completed and we have the result, people would say that, the whole question is outdated already, we don't use that kind of therapy anymore. But that's life, and I don't see how we could avoid that. So you need adequate sample size, you need a standardized intervention, you need a comparator that is really a realistic alternative to the new treatment. The outcomes must represent the real goal of the intervention, and you need to have substantial resources in order to conduct one. The strengths, they are potential, they are not automatic, they are not always there, because as I said, randomization alone is insufficient as a guarantee of high quality. All procedures within a randomized study can be conducted either well and appropriately or badly when they would introduce possibility of bias. And just the fact that the study is randomized doesn't mean that it's good science. There is an excellent book by Ben Goldacre called Bad Pharma about the ways you can tamper the results of a randomized trial. Okay, so just to sort of summarize, for a high-quality randomized study, you need to have clearly relevant inclusion criteria. You need proper randomization, meaning that the outcomes are unpredictable. You need large enough study population, providing sufficient power, even allowing for dropout and loss and non-compliance. You need a standardized intervention for the vast majority of the subjects. It's not very rarely all will receive it nevertheless. Blinding should be maintained if using subjective outcomes. Good adherence to the intervention is needed. Comprehensive follow-up, that's something I haven't covered in such detail, but you should have objective and standardized measurements. You need to analyze the data in accordance with the allocation, meaning that using the intention to treat principle. The couple of words of warning, reporting may be biased, and therefore we nowadays require pre-published analysis plan. Subgroup analyses are commonly reported even if they are not specified a priori in the plan, and they are very often incorrect because the subgroups are not randomized and the power is often limited. You need enough data, enough events to come up with a firm answer, and poor compliance and high dropout are issues that are often there for randomized studies. And another question is trials that are terminated early because of benefit or because of harm, lack of benefit, and this may be problematic for instance, a systematic review. Okay, so final slide to summarize. Randomization is a means of isolating the effect of an intervention from the complex array of cofactors. That is why we need randomization. Randomized trials do produce valuable knowledge, but they are not always high-quality science. So they have the potential to do so. Properly conducted randomization, the chief virtue is by creating comparable groups at baseline. And the main challenge for randomized trials is keeping the comparability. So in order to come up with a good randomized trial, you need to have a good design, you need to have conducted well, you need to make the measurements well, you need to make the analysis properly, and then you can provide a good answer to the research question. And just some books. Thank you.
Thank you very much. Спасибо. Один вопрос.
Thank you very much. So now you can ask one question if you have any. I will speak Russian. I have a question about subgroups. So subgroups should be randomized, just like other groups. So you had a slide about subgroups, one of the last ones.
So, yeah, еще вопрос по какие подгруппы? So subgroups should they be randomized as well?
Where was it here?
We should randomize.
Yes, subgroup analysis are not truly based. Yeah, should be random.
What about subgroups?
No, typically subgroups are not randomized, and nearly always they are not randomized. So, for instance, if you don't see anything in all of the patients, and then you'd start, oh, but there's a large effect in women but not men, or the other way around, or older patients but not younger patients, or whatever. You can come up with infinite number of subgroups, and for me it is fishing. It is post hoc analysis, and there's a famous trial analysis also showing that in even if you analyze pooled trials by horoscopic sign, you will see differences. Okay, if there's only the Gemini and Libra that don't benefit, all others will benefit from this intervention, which clearly doesn't make any sense.
It's not a problem.
It's not. It is a problem. It is a problem. So you should avoid subgroup analysis. They are exploratory. They are not trustworthy until they have been confirmed elsewhere.
But we can do it. Can we do it?
Of course you can, but whether you should is a more tricky question. And I would be very cautious about conducting and reporting them very carefully that, okay, this was completely post hoc, exploratory, and the results suggest something, but as we don't have the power, this was not hypothesis that was specified in advance, therefore we should interpret them very cautiously.
Can we call it randomized study?
Sure, if it's conducted within a randomized study, but then it's subgroup analysis.
Thank you. I think we'll discuss it.
Okay.
Yeah. So, thank you very much.